In studio, we have Mike Gogarty. He is working on a different kind of heart procedure. And Mike, just give us a little bit of background on yourself. How did you get to this point?
I'm older. I'm a non-traditional student. I have a bachelor's and a master's in mechanical engineering. I've worked in industry for about 15 years and worked in aerospace and biomedical engineering. And I decided I wanted to come back to get a PhD so that I could teach at a university. I really enjoy biomedical engineering, so I actually work full-time in the mechanical engineering department, and then I'm also working on a PhD in biomedical engineering in the cardiovascular and biofluids laboratory.
And where'd you do your undergrad?
I did part of it at the University of Colorado Boulder and also at BYU in Utah.
All right, we'll forgive you for that minor transgression, but
I only was there for 2 years. I didn't like it either.
okay, okay. You're still one of us. Tell us what your research exactly is in that you're doing right now.
Okay. So in the laboratory I work in Dr. Prasad Dasi's laboratory. It's the cardiovascular and biofluids laboratory. Our main focus is basically all areas of cardiovascular and biofluid mechanics, and we focus on understanding how we would go about designing things like heart valves or how a heart develops and things like that. My area of research is to look at the methods and the process of replacing a diseased aortic heart valve. Current gold standard is an open heart surgery. The open heart surgery is exactly what it sounds like. They cut open the chest, remove the sternum, pry open the ribs, stop the heart, they put the heart on the blood on a heart-lung bypass machine. Then they stop the heart and then cut it open, remove the old valve and suture in a new valve, and then suture up the heart. And at that point, they try to start the heart again. If they're successful, then they continue to close the chest, and then the surgery is complete. The time involved in that recovery time is about a year. People have been known to get very depressed because they can't quickly return back to activities that they're used to. The survivability rate is about 70%. 30% of the people within the first year that participate in that surgery will die because of the invasive nature of the surgery. And those are the people that have been selected to or been told that they're healthy enough to participate in the surgery. So the goal of my research is to develop or investigate the process and the methods to do what's called a minimally invasive transapical aortic valve replacement off pump. And what that means is that, first of all, we're going to do a minimally invasive procedure. So we're not going to do something that is opening the chest or anything like that. So what we plan to do is make a hole about the size of a nickel, the diameter of a nickel, just below the left rib cage, the last rib and the rib cage on the left side, and go into the apex of the heart, which is kind of the pointy part of the heart that's on the left side of the body. We go through the apex with a tool, and that gives us pretty much a direct shot into the aortic valve. What we want to do is keep the heart beating while we do this procedure, so there's no risk of having to restart the heart. And so the first thing that we would put in place would be a temporary valve. The temporary valve would then function as the aortic valve, allowing the heart to continue to function as a pump while we proceed with removing the leaflets in the aortic valve. Many times the valve is diseased or it has calcified nodules in it, and so we would remove the leaflets and the calcium and prepare the site. This is something that's never been done before. They don't typically prepare the site unless they do a full open heart surgery. There are some methods where they go in and they are minimally invasive, but they actually just put a valve on top of the calcified leaflets, which can lead to problems where blood can actually flow past the valve. The valve doesn't open all the way, and actually valves have been known to migrate up into the aortic arch, and in some cases closing off the two sinuses that leave the aorta. And so the goal would then be to implant a new valve, remove the temporary valve, suture up the apex, suture up the hole in the thoracic cavity, and that would be the end of the surgery.
All right, so putting in a new valve, what do you make a valve out of?
So there's this is one of the reasons this is also a very attractive method of doing this. There's currently only two options for a valve replacement, replacing an aortic valve. You can have what's known as a bioprosthetic or a completely man-made prosthetic or valve. And the bioprosthetics are partially man-made and partially a biological material. Typically, it can be made of pig pericardium, which is the sac that goes around the outside of the heart in the thoracic cavity, or it can actually be made of pig leaflets from the aortic valve of a pig. The pig heart is actually a good analog to the human heart. The problem with that type of a valve is it typically only lasts about 8 to 10 years. And so, for instance, if you're a younger person, a child or a young adult that has a heart disease or a problem, then your options are essentially to have open heart surgery several times throughout your life if they were to put in a bioprosthetic, because it's only going to last 8 to 10 years. And there's many people who are born with congenital heart defects. It's not a diseased heart or anything like that. It's just a defect. And so they would expect having to have multiple surgeries throughout their life, multiple very invasive surgeries.
Right.
So the other option is the completely man-made valve. They last forever. They're great. The problem is they damage red blood cells. They actually make the red blood cells start to form clots. And so if you have that type of valve put in you, then you have to be on an anticoagulant or a blood thinner for your entire life, which means your blood will not clot if you're in an accident or a surgery or anything like that. And so you could actually bleed out and die as a result of that. And so neither of those options is great. So if we were able to develop a minimally invasive method, if a valve needed to be replaced multiple times, it wouldn't be as traumatic to the person or as expensive. Recovery time wouldn't be as long. Hopefully, and there is research going into valve design. We hope that eventually we'll come up with a valve that will last a long time and not damage the red blood cells. That's actually research that's going on in our lab. But until that happens, if we had to do multiple surgeries, this would be a much better option.
Right. So with your method, you're coming from down below. What does the development process for this method look like? Did you just like dream it up, or is this trial and error? Like, did you play Operation and then
Yeah, that's good. It was a little of all of that. So we did a little bit of trial and error, a little bit of sitting down and trying to figure out how to do it. My advisor, Prasad Dasi, had some ideas. We had a master's student and another PhD student kind of play around with some things. They were doing some additional research, and he just made some suggestions, and they worked on it. So they did kind of an initial prototype. The development process is we really need a system that allows us to design or try out ideas. Obviously, we can't go to a person to try out the ideas. That's frowned upon. Even on animals, that's expensive, even though, and we don't want to just needlessly kill animals as we're testing things out. So what I've created or developed is a system that is what's called a passive beating heart simulator. And it's a system that allows me to take a pig heart, which is, again, a good analog to a human heart, and it actually allows me to set it up similar to what we would experience during the surgery and test out ideas and designs and things like that as we have them. Once a week, we usually go and collect pig hearts from a local butcher. They slaughter pigs on a regular basis, not for us, but they let us have the hearts for free. They've been super helpful. And I take them. We have a way of storing them. We can keep them useful for up to about 2 to 3 weeks. We keep them in our laboratory. And then the system essentially is a large tank. We use a saline solution or water, and we submerge the we do some preparation on the heart, and then we submerge the heart into the tank. And this system actually allows me to control the heart rate, control the amount of fluid that flows through it. So, for instance, a typical adult does about 5 liters per minute of blood flow. And so we aim for that. If we're trying to do something on a smaller heart or do a child's heart, that would be about 3 liters per minute. So we can adjust that. We can also adjust the blood pressure, and we can do this all real time. We just make adjustments. We monitor the results. We can set it up to basically any physiological condition that a person would experience. So even if they had a diseased heart, or they had hypertension, high blood pressure, we can do that type of thing, and set it up to mimic that.
And how do you mimic that?
So the system is, like I said, it's a passive beating heart simulator. So the way it works is we have a pump. It's a diaphragm pump, which is just essentially controlled by putting pressure on one side of a diaphragm, and it ejects fluid out the other side. And that fluid then goes into the left ventricle, and there's an adapter that I designed and developed that we suture to the side of the left ventricle and the wall of the left ventricle. And that goes through the left ventricle, and what happens is the heart's submerged so that the fluid goes into the left ventricle. It pressurizes the left ventricle until the aortic valve opens. The aortic valve opens. The fluid that leaves the aortic valve passes a pressure transducer as well as a flow meter, and goes into what's called a compliance chamber. And I'll explain these things as I go back. Goes through the compliance chamber and through an adjustable resistance, and then back into the tank. When the pressure inside the left ventricle drops, the fluid is actually drawn back into the left ventricle through the mitral valve. And so the heart works very similar to it would sitting in your body, with the exception that we are not having the heart actually contract and expand. But the flow characteristics are similar, are very close, and also the physiological conditions are very close. Now the way we do this is we have a pressure transducer or a pressure sensor inside the left ventricle, and then one outside the aortic valve. We monitor those pressures. We use that to calculate blood pressures, like systolic and diastolic pressures. And then, as I mentioned, there's a flow meter. The flow meter tells us we make adjustments in the system, and the way we adjust it is the resistance valve. It's just a valve. We adjust the flow rate. And the important part past the flow meter is a compliance chamber. Your body is not made up of rigid tubes. And so all the arteries and veins in your body are very flexible, and they actually do a big part of the pumping. The heart will pressurize the arteries, and then as soon as the aortic valve closes, they kind of squeeze the blood back through the system. And so the way we simulate that is just a chamber that we fill with fluid, and we can adjust the amount of air or fluid in there to adjust the rigidity or the compliance, so to speak, to make it similar to physiological conditions. And so that entire system, just as it operates, we monitor the pressures, we monitor the flow rate, and then we make adjustments at certain points in the system. We can adjust how fast the pump pumps. That's our heart rate, and that also controls some of the flow. We can adjust the resistance and the compliance, and all those things affect different things like the blood pressure, the flow. It's kind of all interconnected. And so we basically kind of just adjust things until we get it to the setting that we would like, and then move on to to do some type of testing.
All right. And does this model, does it simulate the actual surgery, or is it just just to test how the valve works?
So since my research is not focused on valve design, so it actually is similar to how we would be doing the surgery. So for instance, on the aortic side, or the side of the where the fluid comes out of the aortic valve, we have a camera. So we have a small camera that we can watch the aortic valve operate. The reason we have that there is not only to watch the valve, but also that as we try out instruments and designs, we'd actually be able to get an idea of how well they're working. And so, for instance, let's go back to the instrument where I said we wanted to do a temporary valve. Well, we would do two things. One, we would watch through the camera to see how the tool goes in place, and if the temporary valve or the tool that we use positions the valve where we'd like. So that would be something that would be experienced during surgery. And then once we put the valve in place, we want to see if the pressure sensors and the flow and everything stays at the physiological conditions before we had when we were just operating it off the aortic valve. And so the camera and all the sensors allow us to monitor things. And it's kind of interesting when I was doing validation testing. So I went through to validate all this, and I took 7 hearts, and this is part of the paper that I've written that we're working on getting published. There was one heart that I couldn't quite ever get set up. The conditions would never adjust to normal physiological conditions, and I thought for a long time that I'd done something wrong. And then I started to look at the heart and notice that the mitral valve had about a 2 to 3 millimeter diameter hole in it. And so it was actually a birth defect in the mitral valve in that pig. And upon further inspection, you could see that the heart was a different shape. It wasn't typical shape. But it was very interesting that the system was able to tell me based on the feedback from the system. I looked at it and I was like, these aren't physiological conditions. What's going on? And I was able to determine that, something was wrong. And so the system allows us to see if something's wrong while we're trying to do a surgery. And so the temporary valve, if while we're doing that surgery, something were wrong, we would be able to see it.
We've gone over what you're doing, how you test for it. What steps are left to really implement this and complete it?
Okay.
How far are you?
Probably about 1/3.
Okay.
1/3 of the way. So, the first part of the system first part of the process was to develop the beating heart simulator. There's some refinement that I'm working on right now with that just to make some improvements. But in general, that's pretty much done. So now we have a system that we can test things on. We've actually tested a couple of things on it already as part of our validation testing. Dr. Chris Orton, who's a cardiovascular surgeon in the vet school, he's on my advisory board, and he was willing to come down one day and kind of try it out. And so there's a couple of things that he's done that we performed essentially on a pig heart in the system. One of them was an annuloplasty ring, and I'm just pointing these out to give you a little background to tell you what's next and to show that we've tested this out. One of them is an annuloplasty ring, and what that is sometimes a mitral valve will be defective, or in often case there is after a heart attack, some of the heart tissue or the muscle will die. When that happens, you get a mitral valve prolapse, which means basically that the muscle is laying on top of the valve and the valve's not working properly. And so they take this little ring, it looks like a D, and they actually put it into the mitral valve opening and suture it in place. And then that holds the opening to the right shape, and then the valve can operate properly. So he actually came down, we did that on the system, and then we're able to operate the system and demonstrate that that works. So if somebody were to have an idea for mitral valve, we could test it out. Then the other thing he did, Dr. Orton has developed a mitral valve replacement. So it's actually a bioprosthetic that he's developed. He's tested it, or is testing it on animals, I think now. And so we took one of his valves and actually put it in place, went through the whole process, and we're able to demonstrate that the system can use that. So the reason I point that out is the next step is essentially trying things out on the system. So the next two things that I want to test, that I'll need to test out, are first a temporary valve. Once we enter through the apex, if we do anything that interferes with the aortic valve, then the heart really won't work as a pump very effectively.
Right.
And so we need to make sure and have a valve that will allow the heart to continue to operate. So we'll first deploy a temporary valve. What that looks like, don't know exactly yet. I'm actually in the process of mentoring 2 senior design teams in mechanical engineering department who are working on a design of that. And then, so they're coming up with an idea on something that they want to try out as a temporary valve. So that would be deployed first, and then the next part would be something of a cutting tool. And so we need a tool that would go in and cut the leaflets out. And there's a lot of challenges with that specific procedure. For one, you can imagine you don't want any debris to float through the body. We have the valve pumping, or the heart pumping, and so it's anything we let go of or comes off will then flow through the body, which would be dangerous.
Right. And that was one of the problems with one of the valves in the gold standard.
Well, it's actually
with the coagulation, right?
Yeah, similar, but the coagulation is actually the blood cells are damaged or activated. It's called lysine.
Mm-hmm.
And they actually send out these little tendrils when they come in contact with a completely man-made prosthetic, and it will actually create blood clots. And so it can actually clot small capillaries or anything like that. You can end up with blood clots throughout your body. In this case, what I'm saying is if we cut off something, we got to make sure that we capture it, that it doesn't go floating through the body. One of the problems, so that's one problem. We'll need a valve, but we'll also need somewhat of a filter, something that's going to catch anything that might go downstream. The other issue that's more difficult issue for a variety of reasons is many of the heart valves are diseased. And by that I mean they're calcified, which means they have calcium nodules that are 2 to 3 millimeters in diameter, could be smaller. The interesting thing about this, sorry, go ahead.
And what does calcification do to a valve?
So it just makes it so the valve cannot open and close correctly. So if you can imagine taking a valve that is very flexible and then adding calcium to it that's very stiff and rigid, it can actually make the valve very stiff and rigid, so it may not close properly. Or the nodules actually could be on the edges of the leaflet, and if you can imagine trying to close something that normally seals and keeps blood in place, you have two pieces that come together. Now they can't because they have bumps on them, and so the bumps prevent it from sealing, and blood can continue to flow through there. So there's a variety of problems. The thing that's really interesting about the calcification, which I just learned about last year, well, maybe it was this spring, but Dr. Fullerton from the University of Colorado Denver Medical Center came up and gave a presentation. He's doing research on this calcification. Many people thought that the calcium was actually just collecting or forming on the leaflets. And so that would kind of lead you to believe, well, maybe we could just pick it off, fling off the little pieces and fix the valve. But it's more invasive than that. It's actually what he's determined is that there's some type of stress or something that happens in the body, in the heart, stress to the cells inside the leaflet, and they actually then they change their phenotype, where they basically change and become a different type of cell. And so they become calcium cells. And so now you have a calcium cell embedded in the tissue, and it starts to replicate itself. And so the nodules are actually embedded in the tissue. So the cutting tool actually has to be able to cut through calcium, or actually cut and remove the calcium nodules without breaking that apart and letting particles float downstream, or we have to be able to collect them. Mm-hmm. And there can be many, like 20, 30, even more of these 2-3 millimeter diameter nodules all around the outside of the leaflets, where it connects to the aorta, around the annulus, which is where it connects around the aorta. So there's a variety of places in it, and so it can make it far more difficult. So another issue we had that I've been working on is we needed a way to simulate the calcification. And since everybody had tried just running high calcium solutions through a heart thinking it was going to deposit, there isn't a method to do that. So I have an undergraduate student that's working with me, and he has I came up with an idea, and he made the idea work. So of being able to basically embed calcium nodules inside the leaflets. And so we believe that that'll be a big step into simulating that, and we'll actually be able to do some research just along those lines, but then also it will help us develop a cutting tool that's more realistic or work in more realistic conditions. So,
Okay. So
so those are the other
problem
2/3.
right. Okay, those are the other two thirds.
Temporary valve and a cutting tool, most likely.
Nice.
Yeah.
That's cool. Uh, so obviously, as you kind of hinted at, there are some issues with inputting this valve. We've got the calcium nodules. What other difficulties might you encounter in just kind of exploring this operation that no one's really done before?
Right. Well, some people have done, so let me give you a little more background. There are some minimally invasive techniques out there, and some people have explored the transapical method. But by and large, nobody's done anything to prepare the site. So the gold standard is going in and stopping the heart and going in and actually cutting out the old calcium and the old valve and everything like that. In any of the methods currently out there, there's nobody out there preparing the site or getting it ready to put a valve in. They actually just put the valve on top of the old diseased valve, and so it's just basically expanded on that. We want to get rid of that. We want to prepare the site. So some of the challenges that we haven't simulated right now that will be part of the surgery is this is very different than any other minimally invasive surgery. The most minimally invasive surgeries take place in a knee or somewhere in the thoracic cavity. Inside the thoracic cavity or your chest area inside there, the way they do a minimally invasive surgery in that method is they actually just inflate you like a balloon with nitrogen. They typically will put in 4 holes into the chest, and it's kind of brutal. There's just actually grommets, like a tent grommet. One of them is a valve. One of them is a place where they inject the nitrogen, and 2 of them are typically access for the surgeon. And so it's holes to the thoracic cavity. But the problem is that area is all dry. It's all nitrogen. You can see, you can put a camera in there, and you can look around. When you're in the heart, everything's in blood. And so you have red blood flowing by. You won't be able to see anything. And so at some point, I expect that this will probably be a future student. We will have to explore how to use like ultrasound or some method where we can actually watch the surgery, because when they do the surgery, they won't have a camera, and even if they did, they wouldn't be able to really see anything because of the blood. And so that's one challenge. Another is, since the heart is still beating, we need to be able to insert tools, for one, of the correct size that won't damage the heart permanently, and 2, blood would squirt out if we didn't have, because the heart's pressurized, we need to be able to seal the opening even while we're doing the surgery. And people have been able to do that, but it is a challenge. And for my system, we need to be able to kind of monitor whatever we try out. We need to be able to see if things are leaking, so to speak. So those are pretty big challenges.
Okay. Excellent. So those are the challenges, and obviously, as you come to them, you'll find solutions for them. As so far, what do you think? What do you think your chances, given that everything's successful, what are the steps to take to make this the new heart surgery? How do you get taken seriously in a place where there's already something called the gold standard?
Right. Yeah, that's true. Yes, we want to be the new gold standard. So first of all, the simulator needs to have good physiological conditions. And by that I mean there's certain pressure curves. So if you look in a book, say a physiology book, a physiology book can show you the shape of the pressure, the aortic pressure, the ventricular pressure, and it can show you essentially how the flow curve. So with my system, that would be the first thing, is we need to be able to mimic those or basically come very close to those. And so those are actually some of the modifications that I'm working on right now, is to get closer and closer to those. So that's a big step. If we can do that and make it so that it looks very physiological, then people are going to say, okay, that's good. That's a good simulator. And now, once we get a paper published on that, we can refer back to that and say, okay, we use this simulator to develop this tool. And so then they won't focus anymore on the simulator. They're going to say, okay, now let's look at the tool that they developed. And they can refer back and say, well, look, they kept physiological conditions while they were using this tool, and their simulator, the data from their simulator worked well. And so that's going to be a big part, and that's something I'm working on refining right now. In the paper that I've written, we have a lot of data, but I need to make some refinements. So that's a big first step. And then the next, really, currently I've only come across one paper, and there might be some more. I just haven't done extensive research on it, but we've only found one cutting tool. And the interesting thing about that particular cutting tool and the paper was, when I initially saw the title, it said transapical, and I thought, oh, great, this will be a good one to look at and see what they did and kind of get some ideas. And the reality is they did what's called a partial sternotomy, which means they went in and removed part of the rib cage and the sternum, and that gave them access to the heart. They never really did a complete minimally invasive method. And so it was kind of not a very good title for their paper. But anyway, so it was good to look at that and see what they did. They had some good ideas. They had a very simple design. I don't think it has to be terribly complicated. We just have to take care of the challenges that we discussed. We need to be able to cut things cleanly, don't do any damage, not leave any particles or anything floating through the bloodstream, those type of things.
And in a field where so much work is noticed by merit, just the process of being a doctor, you have to work a lot before you're taken seriously enough to, be allowed to practice. I can only imagine how hard it is being someone who's relatively unknown in this field, to have their research implemented.
Yeah, I mean, one thing I've run across is surgeons are very skeptical. Some surgeons, let's not say that. I won't use a broad generalization.
Yeah, don't
But there are some surgeons that are a little skeptical of engineers and engineers' approach. And we've been working with the Heart Center of the Rockies. We go to meet. We try to meet with them. They do a journal review. I've actually done a presentation for them to demonstrate where I'm at. And so we've kind of been working with them, and so that helps us to be plugged in with their point of view, so that we won't do something that's just totally crazy or won't be accepted. So it's a really good thing is to be kind of plugged into the surgeons. A lot of the papers out there are actually by cardiologists, and they have a lot of good ideas. And so, but I think it's really good to marry the two, to put the engineering and the surgeons together. And just to give you an example, I mean, there hasn't been a lot of development in heart valve design. There has been development, but actually in about 50 years, not any huge significant changes. There's been some incremental changes, but they still damage the red blood cells. That's always been a problem, and that hasn't been fixed. Now, my personal belief about that is that's because a lot of the surgeons don't understand why. And the engineers who have designed the valves haven't ever taken the time to figure out what keeps it from damaging the red blood cells. What makes a good heart valve design. They just basically said, we need a valve that opens and closes. The very first, or one of the first valves, was a ball and cage. It was just a cage and a ball rolled back and forth, and that's how it worked. Wasn't a very good design, but it worked. And so we have a PhD student in our lab who is actually asking those questions. And so it's the same type of approach. Is instead of just doing surgical tools, we're actually saying, well, can we do surgical tools that won't damage the heart, that will allow us to prep? What's going to make a good tool instead of just saying we need something that cuts a valve out? And so we're actually looking at it from more engineering standpoint instead of just the surgeon's standpoint, which is I need something to cut this out. So I think it's a good marriage of the two, is to have working with the surgeons, the cardiologists, and the engineers, and we can get a lot of really good ideas. I also think it's extremely beneficial, our program, to be linked with the vet school.
For sure.
Yeah. Well, most biomedical school programs are linked with a hospital, and they don't let you test things out on people. Whereas here, we have opportunities to actually go and do animal testing or watch surgeries and things like that typically we might not get to take part in a, where we're hooked up with a human hospital. Plus, we have just some outstanding people at the vet school that are, tops in their field, and they're just great to work with. So very knowledgeable and helpful.
All right. As we enter almost the 40th minute of our interview, we just have one more question to wrap up. What's next for Mike Goharty? Like, after this, you said you're going to be teaching?
That's my goal. I wouldn't have come back to school to just get a PhD. So I came back so that I could teach. So I hope to be able to be a professor at a university, do research, and teach. So I love the biomedical field. It's just great to work in it. It is very fascinating as an engineer to be able to work with biologists and microbiologists and physiologists, and just a lot of stuff for me to learn. And I really enjoy that. I loved that in industry when I worked for a biomed company. I really enjoyed learning all of those things. And so I think it's a fascinating and very exciting field. And so I hope to be doing some instruction and research in biomed and maybe some mechanical engineering. But yeah, I want to teach and do research.
And as far as the field of heart surgeries and development of heart valves, where do you think that'll go next?
Well,
this might be a little controversial.
No. I personally, like I said, we have research going on in our lab. I mean, really, the big focus seems to be trying to figure out how to stop damaging red blood cells. If you can really understand what makes a good heart valve design, then hopefully we can come up with materials or design or whatever, really try to figure out what's damaging the red blood cells so we can create a valve that won't do that. And then if we couple that with a minimally invasive method, that'll be huge in the cardiovascular field, because you'll now have a valve that you can put into somebody that they don't have to be on blood thinners. Hopefully it'll be 100% man-made so that it will last forever and they won't have to have multiple surgeries. And the only surgery they'll have will be a minimally invasive method. There will still be children who have developmental or congenital heart defects. They will have to have multiple surgeries, and so then the minimally invasive method would then be a better way for them, because their hearts grow, and the valves, there are no valves out currently that grow with the heart, and so the only way to deal with that is to replace the valve. Now there are people trying to look into having a valve that sort of grows with the heart, but that's kind of a big deal, so that'll be down the road a ways. But if we could do those two things, that would be huge milestones in the cardiovascular world.
All right. Well, this is 90.5 FM KCSU Fort Collins from Colorado. My name is Tyler Stupp. I've been